Welcome back, students. Today we are going to discuss Unit 5, and today's topic of discussion is photosynthesis. Photosynthesis in higher plants. So, before we start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. In today's video lecture, we are going to discuss these points: photosynthesis, early experiment related to photosynthesis, type of pigments in photosynthesis, what is light reaction, what is photophosphorylation. Okay, so one by one, we are going to discuss these points. So, as you know, that all animals, including human, rely on plant for their food, right? So, why? Because green plants are autotrophs, they are able to synthesize their own food, right, through a process known as photosynthesis. Other organisms are called heterotrophs because they are dependent on green plants, right, for their food need, right, for their survival. And green plant actually utilize light energy of sunlight and create organic compound through a physico-chemical process which is known as photosynthesis, right? So this photosynthesis we are going to discuss in today's video lecture. So photosynthesis is essential for our life. Why this is essential? Because it is the primary food source on the earth. It is the primary food source on the earth. It releases oxygen to the atmosphere, right? As a byproduct, it releases oxygen as a byproduct into the atmosphere, which is further utilized by us for our respiration. Okay, so in this chapter, we will learn about the structure of photosynthetic machinery. What kind of pigments, what kind of other accessory and machinery involved in photosynthesis. So, structure of photosynthetic machinery and the reaction type, which actually convert this light energy into the chemical energy. Okay, so first we are going to learn this structure of photosynthetic machinery, and then we are going to learn the reactions which convert light energy into the chemical energy. Okay, so what do we know about photosynthesis? One by one, let's discuss these points. As we know this, that photosynthesis require first of all a kind of pigment known as chlorophyll, which is a green pigment. Light, right? Light comes from the sunlight, right? And carbon dioxide, and carbon dioxide, right? And this is a typical picture of photosynthesis. You already discussed this kind of topic or picture since class 6 onwards, right? That there is a plant, it absorb water from the soil and absorb carbon dioxide which is present in the air, right? And in the presence of sunlight and chlorophyll, it will produce sugar as a food and release oxygen as a byproduct, right? So what is the requirement? Chlorophyll, light, carbon dioxide, and starch is formed, right? So starch, let's discuss some experiment which shows that starch is formed after utilizing all these three things. So initially some scientists conduct an experiment in which a leaf is covered partially with the black paper, okay, and exposed to the light, right? So testing for the starch showed that photosynthesis occur only in that green plant which is exposed to the light, which are able to absorb sunlight, which is not covered by uh, aluminum foil or any black paper. Okay, so and so sunlight is required for this photosynthesis process. Is carbon dioxide is also required for to prove this. A part of leaf is actually enclosed in a test tube, and that test tube contain KOH. Okay, and actually KOH soaked cotton, which actually what is the function of KOH? Potassium hydroxide. It absorb carbon dioxide which is present in the test tube. Okay, so a part of leaf is enclosed in a test tube. If this is a test tube, okay, so a part of leaf, this is a leaf, is enclosed in a, if this is a, and here there is a cotton ball soaked with KOH, and this is, we just cover it. Okay, so other part of the leaf is exposed to the air. Okay, so after exposure to the light, the exposed part test showing positive test for the starch, while the enclosed part means this part, which are present inside the test tube showing negative result. Means carbon dioxide is also important for photosynthesis or to synthesize starch as a food. Okay. So, this experiment demonstrates the necessity of carbon dioxide for photosynthesis. Okay, clear. Now, let's discuss some early experiment which was actually conducted by some very renowned scientists. First experiment was conducted by Joseph Priestley in 1733 to 1733. His lifespan 1732 through 1804. He conducted an experiment which demonstrated the role of air in plant growth. Role of air in plant growth. Okay, and he actually discovered oxygen in 1774. Oxygen, which is a part of air. Okay, and he observed that burning candle or a mouse in a closed space would suffocate, indicating air was damaged. Means if this is a cold jar, covered jar, and he just burn a candle and put a mouse in this particular jar. After some time, what happened? This candle is not able to burn, and this mouse is also get died. Mouse died after some time, and it shows that closed space exhaust or suffocated, that after some time the air present in this particular area get damaged. But in the same experiment, he just put it a plant, mint plant inside that jar. Then after some time, 6 hours or so on, both candle is able to burn and mouth is also present in a survival state. Means plant is responsible to convert that damaged air into a fresh air. This kind of experiment was conducted by Joseph Priestley. Okay, so he found that placing a mint plant, this plant, in that bell jar allow mouse to survive and candle to continue burning. Okay, so he hypothesized that plant actually restore the air that animal and burning candle remove. Okay, so this was the first experiment related to importance of air and importance of plant. Okay. Later on, another scientist, Jan Ingenhousz, showed that sunlight is also play a very important role for plant to purify air. Sunlight is essential for the plant process that purify air. He conducted an experiment with aquatic plant. He conducted an experiment with aquatic plant and observing bubble formation around the green part of that particular aquatic plant in bright sunlight but not in the dark. Means bubble is only formed during daytime. Okay, but not in the dark. So he concluded that and he also identified these bubble as oxygen and showing that only green part of the plant are able to release oxygen and in daytime, during daytime only. So this experiment was performed by Jan Ingenhousz. Okay, he used a aquatic plant and observed the bubble formation as oxygen. Another scientist, Julius von Such, he demonstrated a plant produce glucose during growth, and glucose is stored as a starch. Okay, he identified chlorophyll, that these chlorophyll are located in the chloroplast region. So he discovered glucose production occur in the green part of the plant. This was his discovery. What he discovered? He discovered glucose production occur in green parts of the plant. Now, T.W. Engelmann experiment. T.W. Engelmann experiment actually showed that importance of blue light and red light. He actually split light into the spectrum component using a prism. Okay, he illuminate a green alga known as cladophora, which is present in a suspension of aerobic bacteria, and he observed that accumulation of those bacteria occur in blue region of the light and red region of the light, blue light and red light region. That aerobic bacteria accumulate only on those area where obviously where oxygen is available. These bacteria are aerobic in nature, means they need oxygen for their growth. So they are able to accumulate only those regions where oxygen is more. So he observed bacteria accumulate in the blue region of the light and red light region. So he described first that the first action spectra of photosynthesis. So he described first action spectra of photosynthesis, which is resembling the absorption spectra of chlorophyll A and P. This is his very important experiment, Engelmann's experiment. Okay, he actually split the light using prism and illuminate that splitted light on the green alga cladophora in a suspension of water. Right, it is a green plant, green algae, these are present in the water, and suspension of aerobic bacteria. So aerobic bacteria are able to grow in blue light and red light. So he described the first action spectra of photosynthesis. Clear? Now, by mid 19th century, it was known that plant use light energy to produce carbon dioxide from to produce carbohydrate from carbon dioxide and water by mid 19th century. So what is the equation? Equation is like this: CO2 plus H2O is equal to starch plus oxygen. This CH2O represent as carbohydrate such as glucose, okay, which is a 6-carbon sugar. Another scientist, Cornelius von Nile, actually tell us about the photosynthesis, that photosynthesis is a light dependent reaction. Photosynthesis is a light dependent reaction. In green plants, water act as a hydrogen donor, and this released oxygen is actually come from the water. Okay, so in green plant, water is the hydrogen donor and it release oxygen. Okay, as in what is the, just now we have read out the equation just previous slide. CO2 plus H2O is equal to starch plus oxygen. So from where this oxygen come? It may be come from the carbon dioxide, or it may be come from the H2O. So actually von Neil informed or described that this oxygen come from H2O only, not from the carbon dioxide. To prove that, he used pulper sulfur bacteria to use hydrogen sulfide, as these are the pulper sulfur bacteria, so they are able to use hydrogen sulfide, means H2S, as hydrogen donor and release sulfur or sulfate. So it proves that, means reaction type is same, but instead of H2O, hydrogen sulfide is there. Okay, and then starch and then sulfur. So this is how he proved that sulfur or oxygen is come from water or H2S. Clear? Okay. So the oxygen is released by the plant come from the H2O, not from the carbon dioxide. He proved that. And he also confirmed it by radioisotope technique. So what is the actual photosynthetic equation? 6 CO2 plus 12 H2O is equal to C6H12O6 plus 6 H2O plus 6 oxygen. Clear? This is the photosynthetic equation. So now next question arises, where does the photosynthetic reaction takes place? So photosynthesis occur in the green leaves and other green part of the plant, right? So primarily it takes place in the chloroplast within mesophyll cells, right? Within mesophyll cells. So chloroplast in mesophyll align along cell wall to maximize light absorption. This is the structure of chloroplast. It is a membranous system, contain grana. This is the lemon color area is known as stroma, where dark reaction occur. These are coin-like structure are thylakoid. Single structure is known as grana. Okay, these are connected to each other with this stroma lamellae. Stroma lamellae, this stroma lamellae. Okay, and this is the matrix stroma. Stroma lamellae, this is the thylakoid. These are the thylakoid membrane, single unit, granum. This is the outer surface, external surface, intermembrane space. This one is the inner membrane. Okay, and this is the chloroplast DNA. Mitochondria and chloroplast both containing DNA. That's why it is known as these two organelle is known as semi-autonomous organelle. Okay, so this is the structure of the chloroplast. Okay, so there are certain kind of accessories which are present over here and participate in photosynthetic reactions. So first one is light reaction. There are two type of reaction, light reaction and dark reaction. So first one is light reaction. This light reaction occur in the membrane system, and in light reaction production of ATP and NADPH occur. Production of ATP and NADPH. This is the light reaction. These are the thylakoids. Okay, after utilizing light and utilizing water from the soil, light reaction are able to produce ATP and NADPH and release oxygen as a byproduct inside the chloroplast. These ATP and NADPH further utilized by the in the Calvin cycle or dark reaction. Okay, after utilizing carbon dioxide, and in dark reaction sugar is formed. Okay, so this is light reaction. Light reaction, what is the product of light reaction? ATP and NADPH. It is a directly light driven reaction, means photochemical reaction. It require light. It require light. That's why we can use the term light reaction. Dark reaction, this dark reaction occur in the stroma. This lemon color or cream color area is known as stroma. So dark reaction occur in the stroma, and in dark reaction, as I just now told you, it is able to in this dark reaction sugar synthesis occur after utilizing ATP and NADPH, which is produced during light reaction. Clear? So this dark reaction is dependent on light reaction product. What are those products? ATP and NADPH, and not occurring in the darkness, means it is not require darkness necessarily, but actually it doesn't require light as well. It actually depends upon the product of light reaction, means ATP and NADPH. Okay. So, and what is the place here? Thylakoid is the place where light reaction occur, and Kelvin reaction or dark reaction occur in the stroma. Now, what kind of pigments involve in photosynthesis? Chlorophyll A, first kind of pigment, important pigment. It actually absorbs light primarily in the blue and red wavelength. Blue and red wavelength, while chlorophyll B, this absorb light in blue and yellow wavelength. Blue and yellow wavelength. So as you can see, absorption rate and photosynthesis they create. If you compare both the graph, this is the peak of chlorophyll A. This is the peak of chlorophyll A. So maximum absorption occur over here and here, means blue and red region. Okay, for chlorophyll B, maximum absorption occur in blue and yellow region. This one is for yellow. Okay, so photosynthetic rate is also occur at maximum speed or maximum possible rate at blue and red light. You can compare the peak. And so obviously chlorophyll A is involved in the maximum absorption of blue and red wavelength. While this xanthophyll, it absorbs light to blue wavelength. Okay, xanthophyll, and carotenoid, it absorbs light to blue, green and violet region. This is the carotene. Big dash dash. Okay. Now, so after comparing both the graph, we can easily analyze that chlorophyll A actually playing a central role in light energy capturing method. This is the main pigment which actually capture energy, followed by chlorophyll B and then xanthophyll and carotenoid. Actually, these three pigments serve as a accessory pigment. Main pigment is chlorophyll A. Okay, so these three pigments expand the range of wavelength that can be utilized and also protect chlorophyll A from photooxidative damage. Photooxidative damage. Okay, so what is the function of chlorophyll B, xanthophyll and carotenoid? They protect chlorophyll A and protect chlorophyll A from photooxidative damage. But main pigment which absorb maximum light is chlorophyll A. Now, what is light reaction? Light reaction actually a kind of reaction which involve to absorb light energy. In this particular light reaction, water splitting occur and oxygen is released during light reaction, and simultaneously ATP and NADPH formation is also occur. These are certain kind of pigments, thylakoids, right? P680, and this is the reaction center. Okay, so energy come from the sunlight in the form of photons. They can jump from one pigment to another pigment, and after jumping they just come towards the reaction center, from where activated electrons actually accepted by the primary acceptor, then plastoquinone, and it then come towards the various cytochrome complex. Okay, this is the PS2 photosystem 2, where this water is split into oxygen and hydrogen ion. Oxygen and hydrogen ion. So hydrogen ion, and from here electron jump very quickly accepted by the primary acceptor, followed by plastoquinone, and so on. So in this particular reaction, formation of ATP and NADPH occur. Okay, so as you can see, light is absorbed by the pigment, water splitting occur, right, and oxygen is released, right, and ATP is formed. So this is known as light reaction. Okay, so in light reaction there are two type of photosystem works. One is PS1, another one is PS2. Okay, so PS1 and PS2 organize different kind of pigments, excluding one chlorophyll A molecule, and these are actually act as a light harvesting complex. Various photosystem PS1 and PS2. So light harvesting complex consist of hundreds of the pigments. These are the pigments, hundreds of the pigment bound to the protein, and these are actually optimized photosynthesis by absorbing diverse light wavelength. Okay, P700 is present in PS1 photosystem 1, while this P600 molecule is present in photosystem 2. Okay, so P700 in PS1, P600 PS2, these are present in the chlorophyll A molecule to absorb 700 nanometer and 680 nanometer wavelength of light respectively. This is the PS1. So obviously PS1 is related to P700. So it absorbs 700 nanometer wavelength of light. Okay, while P600, that P600 or PS2 is able to absorb 680, not P680 absorb 680 nanometer wavelength. Okay, and further similarly, two electrons are very quickly absorbed, delivered toward the primary acceptor, then ferredoxin, then other carrier molecule, and with the series of reaction, one label to another label means one carrier to another carrier molecule, and then another molecule NADPH will produce during light reaction. Now electron transport. Electron transport occur in through Z scheme. So in PS2, which is known as photosystem 2, chlorophyll is absorbed a light at 680 nanometer. Here, this is the PS2, 680 nanometer. Okay, this is PS2. So chlorophyll absorb a light of 680 nanometer, and after absorbing the light, it actually excites electrons to the higher energy level. From this, as it absorbed light, electron is released and it put a higher energy level. Okay, and these electrons then further transfer to the electron acceptor, which is plastoquinone, followed by cytochrome B and cytochrome C. Okay, so these electrons are transferred to an electron acceptor and move through a chain of cytochromes, as you can see over here. After excitement, it will further transfer to plastoquinone, followed by cytochrome B, cytochrome C in a downhill form. in terms of redox potential. Okay, in terms of redox potential. Simultaneously in PS1, electrons from its reaction center, chlorophyll, absorb a light energy at 700 nanometer and becoming excited. These electrons are then passed to another acceptor with higher redox potential, and ultimately these electrons are used to reduce NADP to NADPH and H. So this is how electrons are passed from one level to another level, and this is known as electron transport. Now what is Z scheme? Z scheme is a electron transfer pathway. What is Z scheme? As you can see, this is Z. 1, 2, 3. This is a inverted Z. Okay. So what is Z scheme? It is a electron transfer pathway starting from this PS2, right? PS2 to PS1. This one is PS1, right? Starting from PS2 and then move on through various carrier molecule and towards PS1, right? So Z scheme is a electron transport pathway starting from PS2 to PS1, PS2 to PS1, and ultimately reducing NADP to NADPH. And this is referred as Z scheme due to its characteristic shape on a redox potential scale. Okay, after getting light energy, it will convert into the excited form, right? And pass the electrons from one carrier molecule to another, plastoquinone, various quinone, then prastoquinone, then cytoquinone, iron, which is a iron sulfate protein, then plastocyan, and then pass towards the P700, then again P700, get excited and then transfer the electron to a ferridoxine, which is a iron sulfate protein, and then pass to NADP and then convert it NADP into NADPH. Okay, now splitting of water. So in the PS2 means P680 molecule. In PS2, one molecule of water is split into two, proton and oxygen and electron. Okay, this water get split into proton, oxygen and electron, and this process replenish electron for PS2. It occur at the water splitting complex within PS2 on the inner thylakoid membrane. These are present in the inner thylakoid membrane, and oxygen is released as a byproduct, and which is contribute to the net production of oxygen in photosynthesis. Okay. Now, photophosphorylation. What is the meaning of photophosphorylation? Phosphorylation means synthesis of ATP. Photo means light. So, synthesis of ATP in the presence of light. Okay. So, synthesis of ATP from ADP and inorganic phosphate in the presence of light is known as photophosphorylation. Clear? So these photophosphorylation are of two types, non-cyclic photophosphorylation and cyclic photophosphorylation. Non-cyclic photophosphorylation include both type of photosystem, means PS2 and PS1. PS2, what is the center? P680. PS1, what is the center? P700. Okay, so both the photosystem involved in non-cyclic photophosphorylation, and these are working in a series, and it is connected via electron transport chain, which is known as Z scheme. Just now we have discussed this Z scheme. And in this non-cyclic photophosphorylation, both type of molecule means ATP and NADPH form. ATP and NADPH. While in cyclic photophosphorylation, only one type of photosystem means P700 means PS1 is functional, and electrons circulate within PS1, and it is occur in stroma lamellae, not in thylakoid, and only an ATP is produced in cyclic photophosphorylation, not NADPH. So this is the difference between cyclic and non-cyclic photophosphorylation. It happens when light energy wavelength is more than 680, okay, available, because P700 molecule is active in this particular cyclic photophosphorylation. So this is all about the today's video lecture, which is related to photosynthesis. I hope most of the things has been cleared now. Still you have any query related to today's video lecture, please let me know. We will discuss it later on. Till then, take care, bye bye, and keep learning. Namaskar.